Hi, how are you today? Okay, how was the exam overall? Good. I expect a lot of students to have an ace the exam, I hope. Just kidding, okay? uh, we'd like to continue on where we have left. We just stopped right before the discussion of the mitochondria in the course of the organelles, different organelles functions and structures, right? So now it's time for turning our attention to mitochondria. So mitochondria is the cellular, like a power factory. Yeah, it generates huge amount of energy in the form of ATP. So without mitochondria, the amount of ATP that a single cell can generate out of per unit nutrient like a glucose molecule is pitiful, negligible, very small. But the same glucose molecule can be converted into huge amount of ATP. There is in the general biology textbook, there is the number-wise comparison between the mitochondria, in the presence of mitochondria versus absence of mitochondria, how much ATP that a single glucose molecule can generate in a cell. Yeah, there is something like a 2 ATP versus a 38 ATP, something like it's more than 20 times, I mean almost 20 times as much as those ATP with the same amount of the glucose molecule. So that is revolutionary the scheme of metabolism, of course. So that's why I emphasize that without mitochondria, the eukaryotic cell's evolution would have not been possible, because since once you decide to become a eukaryotic cell, then you should be able to deal with the much higher demand for the energy. You are not a single cell, the small size prokaryote anymore. So that is the key event, actually. So then now, if we think about it, okay, how did this eukaryotic cell obtain this mitochondria? It's been a mystery. So to explain that, there was a theory called the endosymbiosis theory. Symbiosis means living together. A lot of organisms actually establish this symbiosis at many different levels. Okay. We, as a human, individual level of organism or individual level, we establish so many different variety of symbiosis with so many different microorganisms in our guts, right? In the intestine. So actually defining the concept of who are you? Are you really you are simply the one made of your cell only, several trillions of cells, that human cell as an adult human, for example? That is it became some skeptical, because without all those symbiotic microorganisms in our guts, you are nothing. You cannot do anything. If you get rid of some portion of the bacteria living inside of you, you no longer can function normally. So that means actually, who are you really? Shouldn't you some whole combination of entire your cells plus some other symbiotically living or microorganisms together? Because they are actually doing so many wonderful things, including they actually have some kind of a strong communication. This microorganism over here inside of us establishes some specific communication with our brain, and they send the signal, hey, we need some kind of specific molecule, give us. And then the brain receives such a demand, the signal, and then they send through the nervous signal to specific the glands that produces the hormones. And then, okay, produces and the necessary materials are being provided to them, and vice versa. So give and take is a mutually mutually beneficial the relationship. So that's one very good example of symbiosis. Here, okay, so expanding such symbiosis example into the micro scale that we are going to take a look at it now. So internally, at a single cellular level, it says from the beginning of our eukaryotic cells history, not to mention these humans or chimpanzee or some more higher animals, but simply at the beginning stage of this eukaryotic evolution, we actually started this living together thing, symbiosis, with the bacteria, some very strange, mysterious bacteria that happens to be an ancestor of mitochondria, is what this endosymbiosis theory try to sell you. So this the cartoon try to explain this endosymbiosis theory. Here, not only did this cartoon explain the origin of this mitochondria is over here, like here. Okay. But also, this one try to explain the origin of other membranous organelles in our cells. The other groups, do you still remember that names, that endomembrane system? Like the day before, we actually took the last week, when we had the last lecture before the exam, endomembrane system, all different organelles, including like endoplasmic reticulum, Golgi, and lysosome, blah, blah, blah. They are all the endomembrane system called these. They are these. Why they are called endomembrane system is that actually these scientists believed that they are actually the result of a push the cell membrane. Originally, this was the initial, the cell who had an ambition to become a eukaryotic cell. Okay. So I want to become a eukaryotic cell. Okay. Nobody intentionally can have such ambition and then can go to that direction in the during the course of evolution, of course. But just jokingly, if I'm allowed to make a joke like that, okay. So this cell had such an issue, agenda. Oh, okay, I want to become a eukaryotic cell. So I want to become larger, and I need some internal organelle. How am I going to be able to provide them? So somehow the external outside membrane, cell membrane, was somehow being pushed inside. So if you push the surface, sometimes you can have some internal folding. Okay? So these, after keep pushing it, so internal folding, these little internal folded ones still have a boundary, same boundary made with cell membrane. That's why, that is the reason why those endomembrane system consist of exactly the same chemical type of cell membrane, just phospholipid bilayers. Okay, that's what they say. Believe it or not, it's totally up to you. Okay. So even the nucleus, if you keep pushing it, the central area of being pushed inside, folded region somehow, okay, meet together at the center to become encircle the central DNA area chromosome. So that became the nuclear envelope. Okay, so at least it makes sense. I mean, whether we have no idea, we have no way to prove this. This was the course throughout the eukaryotic cell, all those internal membrane system has established. Okay, but anyway, okay, fine, whatever. And then what about this mitochondria? Also, then the origin of mitochondria is also like along the line of this being pushed inside It's called the professional term. They call this an invagination. This kind of a membrane inward folding invagination also occurs during your regular developmental procedure when you first early stage of the development in your mother's body, actually. So anyway, this invagination explains the other organelles in eukaryotes. But this invagination theory, internal invagination theory, cannot explain the origin of mitochondria. Why? Because mitochondria, structurally speaking, is so much different. They are different class. Hey, I'm different from you guys. One thing, they are made up of 2 layers of a different cell membrane. It's different from the 2 layers of a cell membrane, the nuclear membrane, chemically speaking. So you cannot simply claim that, oh, this mitochondrial membrane was also the result of this keep something pushed inside. So originally the external surface of your cell membrane became the mitochondrial membrane. Then everybody will laugh at, who are you? Are you crazy? Because that doesn't make sense. So then, okay, in order to convince all those doubters, scientists have to come up with another lie. What kind of a lie can we make? So that's how they just came up with this so-called endosymbiosis theory. So it's a joking when I said a lie, okay? We don't even know whether it's a lie or not, but it's a plausible theory. That's why it's still up in the textbook, the endosymbiosis theory. I bet some of you even learned that in your high school, biology too, I guess. So, according to this endosymbiosis theory, the mitochondria, the ancestor of mitochondria, were actually independent, free living bacteria. But this bacteria has some uniqueness in terms of their capability of, they can utilize the oxygen. So remember that when the environment of Earth has drastically changed eventually due to the increased level of oxygen, now the high level of oxygen at the time became, for the majority of most of the existing, at that time, existing organism, that was totally disaster. So majority of them had to extinct because of such high level of oxygen. Oxygen must have been toxic, extremely toxic to them, because they have never been exposed to such a obnoxious material. What are they doing? Because oxygen is highly reactive. So the existing back then organism, they must have had such a hard time to dealing with. But during such course, during such turmoil, probably some of them luckily found a way to somehow evolved, able to evolve to be able to utilize those oxygen. There's a new material. Oxygen is such a new thing. Instead of regarding them as something toxic chemical, okay, was there any way I can utilize this thing? And bingo, they had a jackpot, so that using this oxygen, they somehow developed totally new revolutionary way of metabolism, generating huge amount of ATP. That must have been their ancestor. Okay. Why don't you simply live alone, independently, happily ever after? I don't know. But somehow they came inside to a bigger guy, a larger guy, who was in the course of becoming more complete version of eukaryote. But their journey to the complete perfect eukaryote was not done yet, because of this something is missing, the energy. In order to become larger, having all those other internal organ, okay, to function as a compartmentalization object. Okay, so check, check, check. You check the list, check, 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 but still something is missing, which is how am I going to fund this in terms of the energy? Now, then, one day, all of a sudden, this guy came in suddenly from outside. This is the ancestor form of mitochondria. Usually, the fate, if someone, as an intruder, if someone came inside of cell, what is the fate of this intruder? Total destruction. Digestion and destruction should be a normal fate, but still it's a mystery why this guy was not destroyed, but rather simply remained unharmed, and then they somehow ended up signing contract between this host and the visitor. Okay, from now on you and I will do this. Okay, I'm going to provide this and you're going to provide that. It's kind of an agreement, and then they live together. It's the end of the story. It's too much of a fairy tale. Who can believe this kind of BS? How can this mitochondrial ancestor, whatever, was spared? It's not destroyed. Nonsense. But you are looking at this, actually, the presence of this guy inside of eukaryotic cell, which you cannot deny. And how else are you going to be able to explain that? If you don't believe this endosymbiosis theory, then are there any alternative version which is also plausible? Then we will take it, but none yet. Okay? So, somehow must have been a huge blessing for this eukaryotic cell wannabe, because instead of killing this mitochondria, allowing this mitochondria even replicate on their own so that they can maintain their decent number of copies inside of cell, like some cell, depending on which type of eukaryotic cell in your body, certain tissue cell can have as many as several hundred mitochondria in a single cell. Some other cell which doesn't require too much of this energy demand, then can maintain the fewer number of this mitochondria. It totally depends on the host cell's need. But anyway, multiple copies of this mitochondria definitely will help in the form of this energy, huge energy. So that's why I can definitely say that without this mitochondria, multicellular organism must have been absolutely impossible. Okay. Only after you acquiring this mitochondria, now you can think of, should I have this multicellularity? And that is the reason why you do not see any multicellular organism in prokaryotes. Prokaryotes don't have any mitochondria, but you don't see any multicellular prokaryotes. Only after you becoming eukaryote, now you can think of. It's your choice now. It's your option. Whether still after becoming an eukaryotic cell, whether you still trying to maintain your simpler lifestyle as a single celled, or you can become much more complex. You can enjoy those complexity and luxury as a multicellular organism like human. Is totally your choice, the ancestor choice. But anyway, okay. So that must have been such a blessing. So now here we are. Then the final question is, okay, then where is the evidence? Is there, yes?
이거 수업 좀 끝나서 미안한데요.
That's okay.
그러면 그 많은 생생물 미토콘드리아가 퇴화되어서 자기가 직접 아무리 먹어도 자기는 에너지를 잘 먹을 수 있고 이렇게 대부분의 에너지를 숙주 생물한테서 빼앗아 먹는데 그러한 생물들은
The cost. Listen, as the parasite. If you are a parasite, now you keeping your internal mitochondria, and then you calculate your cost. How much do I need to? I mean, the price. because you don't really need to generate that much energy on your own, because you should be able to suck up all those energy you need from the host. So that's why you ask me and then you don't listen. So such energy, you don't really need them. So that's why some parasite don't have to maintain their mitochondria anymore. So after they acquire this mitochondria, once they decide to, okay, from now on, I'm going to be absolute parasite, then I don't need any mitochondria anymore because I don't need to generate so much energy on my own, so they dumped. So some of those parasitic organisms like parasitic protists don't have any mitochondria. But that doesn't mean from the beginning they didn't have. No, they did have. But only after becoming this parasitic relationship, they decide to discard them. So the evidence, so you can only read them. I don't have to read these together, so I will skip. So now it's evidence part. So there are 3 lines of evidence that can support this endosymbiotic series. One is this DNA thing. Yeah. If the mitochondria was not the independent ones, the originally from the independent living free organism, they didn't have any reason for having any piece of element, the DNA element on their own inside, but they do have. They do have this DNA, and moreover, these DNAs are not linear but in circular. This is more important. So if that was, you can argue, okay, mitochondria have DNA, big deal. If you decide to become skeptical, you can be skeptical to some extreme so that you can still argue that, okay, fine, the reason why mitochondria still have DNA is that somehow nucleus DNA can be spilled outside so that this mitochondria can pick up. Why not? Right. But then that thing is not possible because this DNA is totally different. It's prokaryotic DNA, circular DNA. Remember, all those prokaryotes, bacteria, have this circular DNA, whereas all those eukaryotic DNA are this linear DNA. That makes totally different, and that is evident. That's why now can become one of the evidence for this endosymbiotic theory. They have this circular DNA. And another thing is they do have also ribosomes. So even they can cook themselves. So that means they can make their own protein inside. They have their own gene, and out of their gene they can make their own proteins. Okay, the actual thing what happened is when they came in originally, they brought so many more genes inside of these eukaryotic cells. But then most of them actually have migrated into the nucleus. So in these days, we found the mitochondrial origin of DNA in the nucleus chromosome. But still some of the stubborn mitochondrial genes still remain there, and inside they are doing their own activity in the mitochondria. And to support such activity, these ribosomes are prokaryotic, which means there are 2 different classes of ribosomes, if you remember. Like these ribosomes are actually sensitive to all those antibiotics. This ribosome, because they are prokaryotic. So when I said that earlier that all those antibiotics are actually not going to affect eukaryotic cell because they are designed specifically only for this prokaryotic metabolic interruption, but actually I lied, because inside of us actually we carry these prokaryotic properties. So those properties will be affected if you overdose. Okay. So if you have a higher, much dosage of overdose of antibiotics, then your mitochondria will be affected. So you don't want to do that, actually.
그러면 이론적으로 항생제가 미토콘드리아에 영향을 미치는데 미토콘드리아 이거 사람 미토콘드리아까지 악영으로서 생기는 부작용은 없나요?
So overdose. Antibiotics overdose will be the one. Not all antibiotics will affect this mitochondrial metabolism. There are so many different ways of you can develop the antibiotics, right? So, but there are many kinds of antibiotics targeted to the ribosomes. Okay? Then those antibiotics will be harmful if we have so much because of this mitochondria. Okay, these two are legitimate. These two are all legit examples. Then what's one last example is the membrane thing. They have two membranes, mitochondria. Unlike cell membrane, our cell membrane consists of a single membrane of double dual phospholipid bilayer. But this one phospholipid bilayer, this mitochondria has two, outer and inner, two things. Probably you didn't understand. Maybe I just poorly explained. Okay, one unit of phospholipid bilayer consists of this, like, okay. Excuse my poor drawing. So this will be one unit. Upper, lower, upper, lower. That's why we call this as a phospholipid bilayer, upper and lower. And this is still regarded as a single unit, one. So when I say double membrane, which means you have two of this, one and two, outer and inner, of each of which consisting of this same phospholipid bilayer thing is, so it has a two instead of our cell membrane has one. And more importantly, their chemical property is different. Outer membrane's chemical property is, biochemical property is similar to that of archaebacteria. The inner membrane is, guess what? It's more similar to those of modern present days of bacteria. Although archaebacteria and bacteria, both of them are prokaryote, but fine details are different. And remember, the origin of actual eukaryote, eukaryotic cells, were what? We were all derived from which one? Bacteria or archaea? We are derived from archaebacteria. So how this thing can serve as one of the evidence for this endosymbiotic theory? This mitochondria consists of two things, but one thing is, the outer thing is like us, our ancestor. The outer surface chemical structure is something similar to our ancestral archaebacterial thing, and their inner thing is their original, their own. Is like if we draw such thing, it's just like this. Okay, so this is us that in the course of becoming an eukaryote, and this guy is a visitor, as a mitochondrial ancestor, and came in. The only way this thing can enter is like this, push. And during this push thing, you inevitably end up there. Hey. You end up picking up this portion. So now this thing ended up having entirely encircled with this outer thing, which was once this part. Okay? So, and this part turned out to be archaebacterial cell membrane, and this thing originally similar to bacteria thing, which fits to the theory. So, if you are going to make up a lie, you better make up like this very plausible lie like this. Then people, okay, Still, I cannot really take it, but there's no way I can actually refute this. So if any one of you over here had an idea, okay, I can refute this thing. Because of this and this, this thing is wholly BS. Yeah, I will respect, not only respect that, I am willing to award you, regardless, I am willing to award you A+, okay, if you can come up with such things. So keep trying until before this class is over, okay? Now, so that's the end of, although we have so many other things so we can talk about, but yeah, due to some limitation of this time and many other things, so we should move on to something different. Like now, let's take a look at the cytoskeleton. As the term itself indicate, this cytoskeleton means a skeleton, our backbone system, right? So it behaves a very similar function as our actual skeleton system. But the only thing different is that they are inside of each individual cells. So these cytoskeletons are obviously prokaryotes do not have this type of luxury. No prokaryotes can have this cytoskeleton. This is only eukaryote thing. So eukaryotic cell only can have this cytoskeleton, and they can have so many different functions, including first, thanks to this cytoskeleton, you can define your specific shape as a eukaryotic cell. Different eukaryotic cells have the very distinct different shape. Remember, a neuron, nervous cell, which always have this very, very peculiar distinct shape. Think of your surface cell, like an epithelial cell, have a very specific way of surface. All those different shapes is the result of actually inside of a cell, there is some specific design, the layers of this protein. All these cytoskeletons are maintained by 3 different types of proteins. These proteins' common characteristics is these 3 different proteins' common characteristics are, guess what? If they are going to act as some kind of skeletal things, what kind of property? So let's say this is an entire space is one internal space of a eukaryotic cell. So now you are going to lay many different protein as like cytoskeleton function. What kind of a peculiar common characteristic shape do you need? The round, small, we call that a globular. If a protein shaped like this circle, then we call those proteins as a globular protein, globule. So that's why the antibodies, another name for these antibodies are immunoglobulin. And also you have heard that hemoglobin, those globe, they are all those roundish proteins. Okay? But instead of this globular protein, you better have some long, slender, fiber-like protein. And that's the common characteristic of all these 3 different cytoskeleton protein naming. Like one is an actin microfilament. Look, filament, microfilament, and intermediate filament. Well, actually, one of those intermediate filaments happen to be inside of a nuclear envelope is the one actually causing that progeria. Okay, when you don't have it, the nuclear envelope will collapse, so the progeria develops. If you still remember that progeria. And microtubule. So this is tubule. What's the difference between tubule and the filament? The shape. Tubule is something larger in diameter and hollow, empty internally, whereas a filament is smaller in diameter but is filled inside. So that's exactly what this actin versus microtubule. Actin is a filament, smaller in diameter but filled inside. But microtubules are larger but hollow internally. Okay. So this is an actin. So all of these, another characteristic, the common characteristics of these three different types of the cytoskeletons are, they are all made up through synthesis from smaller individual subunits. Okay. So always that's the way. In the cell, when you try to make something bigger, larger molecule, it's just a matter of your choice whether you are going to this Tesla route or other regular automobile building route. Like if you take a Tesla route, you don't have to have any assembly, but simply you have a large already pre-made frame. Then you just synthesize them. But the conventional way is assembling small part into a bigger form. But cells can only do this route. So whenever you need some big molecule, big thing structure inside a cell, your only choice is you have to gather some small parts, components, and then assemble them. And that's how all these three different big, large, slender filament fibers are assembled. So, which means each individual ball-like round globular proteins get together and assemble these fibers. Filament is called now actin filament. And in the case of, and what about this? What is this? This is one of the typical animal cell, the fibroblast cell. And actually, if you decide to stain only the actin inside of your animal cell using one of the specific the fluorescence dye, then that's how you. So it means your actin filament is everywhere in your cell. And there is a reason why this fibroblast cell has this distinct shape is, okay, big deal. And then what about the intermediate filament? Also, it looks like it's everywhere too. The green thing, in this case, in one particular animal cell, if you stain all the presence of intermediate filament using this specific fluorescence dye, only staining only this intermediate filament, then you end up this, which means they are also everywhere. So they overlap. Actin and intermediate, they just entangle so complex in such a complex pattern. And what about the microtubule? So this is a tubule, isn't it? The inside internally is an empty thing, as it's a larger, and also this individual subunit. Okay, after assembling all this individual unit all together, they ended up this whole big structure, like hollow tube-like structure. And look at this, all these green fluorescence stainings indicate the presence of the microtubule. So in other words, all these three different types of cytoskeleton proteins are everywhere, not only everywhere inside your cells, but they are so highly complexly entangled each other. So probably you don't have any space, empty space to maneuver yourself inside of your cell. So it's filled like all these spider webs. And that is something what we need. Otherwise, we cannot function because this is a summary of what can all these three different types of the cytoskeleton protein can do in terms of their function in a cell. So this is a cartoon of a typical our surface cell, which is called epithelial cell, your skin and your outside covering of internal organs are all made of this type of cell called the epithelial cell. But anyway, the reason why all those epithelial cell has this peculiar finger-like projection is thanks to this actin proteins are standing upright at the region, and also the big highway like all this blue Korean airline color blue, this lines indicate all these microtubules, big highway like things, along which different things are being moved. Okay. So when I said this exocytosis or endocytosis, through which all these different proteins and the component materials are moved from one location to the other, so that eventually some of them are go outside, and some of them ended up in different locations. They all, I just kind of gave you impression of, okay, here, this is one cargo, catch me if you can, like this, like balloon thing. No, it doesn't go that way. In reality, they always go through already pre-made, well-designed tracks, like road-like things, which is provided from the internal cytoskeleton. So two things already I have mentioned. See, all this cytoskeleton protein first can function in providing the particular shape, okay, of cell, both animal cell and plant cell, of course. And second thing is also the functions in this material transport inside of eukaryotic cell. So one material in the form of transport vesicle, remember that transport vesicle, inside of this transport vesicle, some proteins are captured inside, so moving from endoplasmic reticulum and shipped onto the Golgi, and from Golgi and go to the cell membrane. That routes are actually following this exactly already well pre-made the cytoskeletal road. So the motor proteins here, the motor proteins are specific proteins that walk along the cytoskeleton. So these motor proteins, using the ATP as an energy, like carrying each of transport vesicle, and they walk along one by one the surface of cytoskeleton protein. The reason why I mention it is the famous, when you have this muscle contraction, the famous two names, actin and myosin. Have you heard of these two proteins? Actin is, and these two proteins are like work in pairs and allow you to have this specific muscle contraction, so that's the reason why we can move, okay, actin and myosin. Actually, the actin is the same actin microfilament, the cytoskeleton protein, and myosin is this one type of motor protein only working along this actin only. Now in the muscle cells, their functions are reversed. Now here in the case of the muscle, the myosin is the one actually say something, okay, and move. It provides some force, and then actin is being slided out and in, so this muscle can be contracted. Maybe you don't have any idea what the hell that guy is talking about. Fine, that's fine. Anyway, the muscle contraction is one of these, okay, the one being carried out, the function is carried out by this combination of this cytoskeletal protein 2 is only suffice to say at this point. Okay, so these two functions I have mentioned. And what else? What else can I say about the function of this cytoskeleton using this diagram is fixing the location, particular location of a particular organelle is another such a cytoskeleton function. So if there were not for any cytoskeleton the protein and the individual different, the organelle like this, the Golgi over here and ER over here and some other organelles will not be there fixed more and less, but they can float, move freely all around inside of a cell, which is not good for a cell's health. But this long stick-like protein push them and then fix specific individual organelle into on their respective location is another very important function of a cytoskeleton protein too. So this is an example of where the role of this different form of a cytoskeleton protein in your nerve cell, when nerve signal travels along with, this is only the bottom half of your nerve cell. Okay. Then actually those nerve signal is being sent along with this cytoskeleton protein too. What next? In your cell division too. When a cell tries to divide, the function of cytoskeleton proteins are also critical, essential. In two ways. One is this, the red things are chromosomes here. These are chromosomes. Okay? This chromosome had to be separated. Okay. So this is a two cell. One cell is about to divide into two. Okay, eventually. So this cell will split at the center, eventually becoming two different entities. Now, what happened is this chromosome must be separated and go into their respective two different locations. How? You pull each of this chromosome from this protein, which is the microtubule protein. We call, specifically in this occasion, we call this particular microtubule protein as what? Spindle fiber, which functions to pull each chromosome into the opposite location. But in fact, the spindle fibers are simply made of microtubules. So you have to have a normal function of the microtubule. And that's one thing. And another thing that you need to take care of is actual physical separation of this dividing cell at the cell surface membrane level. Okay? How can you accomplish this separation, physical separation of these two cells? By squeezing. Like if you tighten up your waist belt, eventually you may be able to cut your body into two halves. Okay? That's how you do that whenever you do this cell division. Okay? So squeezing, contraction. This is contraction, the activity. Squeezing activity is another way we can view as some contraction. Always, whenever this particular function contraction is up for, remember one particular protein, contraction levers to me. Who else? Actin. Muscle contraction give us to me the actin. This cell contraction, yeah, this is our job too, actin. Come up and they form this type of a belt, specific squeezing belt. Okay, we call this a contractile ring, whatever, but who cares? So that's how you squeeze this thing in and eventually cut them into two. So that's how these two different types of cytoskeletal proteins are involved in cell division. Okay, ignore. And the cell surface hairy-like things also the function of microtubules, but just whatever. Muscle contraction finally is over here. Fine. And also, for example, amoeba and also your white blood cells. Some of your white blood cells can capture and engulf some outside thing. And that's typically so famous in the action of the amoeba-like things. And when they do that, they have this temporary, quick, rapid projection, outward projection of the cell membrane. So rapidly stretch out and grab it and then retract rapidly to internalize this thing. That's how this poor thing, actual microgram, electron microgram, this poor little thing got captured. Now the final fate will be they will be inside and then destroyed as food. Okay? That's how this amoeba live. So this rapid projection outside and then retracting, so that in this way they move the surface thing. We call those things as a what? Pseudopod. Pseudo, fake, pod, so, leg. Okay, so these pseudopods are actually the action of the underneath of cell surface. There are so many different actin microfilaments are laid, and they rapidly synthesize and rapidly destroy, disassemble. Amazing, isn't it? And that's how this projection and retraction is being managed. And not only can you see those actions only in this amoeba, but inside of us, we can also function some kind of cell like the white blood cell. Among many different white blood cells, some white blood cells can do cell eating things. That's how they can function this. That's all thanks to this the cytoskeleton too. Okay. So outside of the cell also there are plenty of different proteins we collectively called extracellular matrix. For example, like one cartoon. So I'm simply just zipping through because we don't really have to pay too much detail, unnecessary details. Only some big picture is always important. So let's say this is a cell membrane, our surface. Then actually outside there are so many different proteins out there too. Some of them actually, the name of some of which maybe even surprisingly familiar to you. For example, I assume everyone knows this name, collagen. Okay. And also some of them, thank you, no, not yet. Proteoglycans also. These proteoglycans are those proteins that help your health in your the cartilage and so many things. So those aged people love to have some extra the supplement of this proteoglycan too. So many those different proteins are actually functioning outside of this eukaryotic cell, and many different important functions are provided by them, including like cushion-like things. Collagen's main function is providing some padding and cushion-like things, collagen, and also stretching, spring-like. It can resist so much stretching stress too. So collagen, that's why important, and that's why a lot of people believe that collagen, if you have a high collagen content on your skin, then you can look much younger, lively. That's why, that is the reason. And also some other proteins are also providing some other types of very important function. And interestingly, all these so-called extracellular matrix, ECM, extracellular matrix protein, including these like collagen and proteoglycan and laminin, fibronectin, whatever, they are all only found in animal cells. Plants don't have them. That's very strange thing. So this is only our privilege, animal as an animal. Okay, so that's something I can all say about. So I have still plenty of time left. So what can I? Are there any other things that I can go over then? Hopefully. Yeah, sure, why not? Okay, so. So for the remaining 15 minutes, probably I can go over some of your all. It's a long time ago, although that your question. It's really my dilemma is there are so many good questions, and actually I wish I could have extra session only dealing with your questions only, and maintain the class as a like only having this question and answer session would be very good way of having this classroom, because there are so many interesting questions I received that only that I feel sorry that I cannot put up every one of these things. So anyway, one question over here is, yeah, among those prokaryotic cells, always like typically, all the prokaryotic cells are much smaller in size than eukaryotic cells, but there are one notable exceptions like this prokaryotic cell called Margarita, actually. It's not Margarita, it's a Margarita, the bacteria, which is a shape like this. Like one of those group, like one extreme, Margarita magnifica or something, like it's huge, it's a matter of a centimeter in shape. Although they have a little segment like things, but they're huge. You cannot really miss. If this is like 2 centimeters, how can you miss this? This is a prokaryotic cell. Are you kidding me? And the really interesting about this type of a prokaryotic cell is actually if you look inside of this kind of big thing, what you can find is actually nucleus-like things, internal membrane. So their DNA, their chromosomes are actually enclosed with their like some nucleus-like version, and they also have some other organelle-like things. So maybe you can go ahead and even say that, okay, this is like in the course of like becoming kind of a eukaryotic version. So what's the point? So although typically all those prokaryotic cells have their own characteristic, typical properties like a small cell size, and so because of that they don't have to have any organelles, but some exception, okay, if one of such exception maintain this huge cell size, and obviously they can have this kind of a eukaryotic-like structure developing, and there is a reason why they do this, but I don't have to go over. Because of this nutritional, the different type of way of obtaining their nutrition allow them to become bigger. But anyway, so before the invention of antibiotics, there must also, before them, there must have so many different mutations resulting in this antibiotics resistance. And then before those availability of antibiotics, what would have they done? Nothing. Obsolete. Yeah, always this thing is always like action and reaction things. Yeah, there are so many different mutations and resulting in some different traits, but if this trait cannot do anything, okay, in terms of your affecting your viability, it will not do anything. So it will have unnoticed. It will have gone unnoticed. But only after we pour our invention, which is antibiotics, onto this microorganism, then now some of those mutations, now is our time to show up, is the thing. So, well, actually, this superbacteria, there was one hope recently, like 5 or 6 or 7 years ago. There was one hope that some of the bacteria living inside of our nose, not every human carry this bacteria, but some, like about half of these whole humans, carry this particular bacteria as they are like a symbiote relationship in our nose. And those bacteria somehow showed this killing ability of the nasty superbacteria, one of the obnoxious, most notorious superbacteria. So just like this, you can somehow discover one of those several natural variants, like from other bacteria or some of the fungi, just like the way we originally discovered and developed the first type of antibiotics. That's probably the way we can deal with different way, trying to think of a different way of developing antibiotics. Okay, are there any other way of amplifying things other than the plasmid? So here, the reason why the upload this question is, let's try to think of what do you mean by this amplify? What do you want to amplify here? Probably when you try to amplify, probably only you want to amplify some specific area of the gene. Then in terms of, so how can you amplify? How can you produce huge number of some specific segment of the gene, that gene of your interest? Only this amplification through synthesis. DNA has to be synthesized. How can you synthesize more rapidly? DNA synthesis naturally is through DNA replication, which is naturally carried out by the specific enzyme called DNA polymerase. Okay, that is the route that somehow invented this particular technique called PCR. This is like amplifying some segment of the DNA using DNA polymerase, the enzyme. However, whatever you do, okay, this DNA, using DNA polymerase and trying to do this DNA amplification can be done in your test tube, okay? But the other way around, if you ask that, is there any other way you can do this thing? Then only you can rely on the natural, actual cell-based amplification. Cell is the one that can provide you such a task of DNA amplification, DNA replication. If you want to do that, then you have to put your thing to the cell, inside the cell. That's why actually this using the plasmid, okay, has been one of the popular choice of this amplification, because this plasmid can be taken up by living cell, and living cell recognize and amplify. One other alternative would be leave this thing to virus, okay. Hey, virus, can you do that? But it's the same thing, because virus also have to infect the cell. So all those virus-made amplification also must be done only inside the cell. That's why it's just the same thing. Okay? So the antibiotics, some kind of antibiotics target these prokaryote specific ribosomes like this. So what's the mechanism of this? Because when I say prokaryotic ribosomes are different from eukaryotic ribosome, actually it's the number of proteins that make up the ribosome is different. And another thing is the type of proteins are also slightly different. It's all what? Once again, structure, shape. So what makes different, critically something importantly different, is a structure. All dictates in biology, all biological function is being dictated by structure. So this different structure can be inhibited by different type of drugs. And we find them through trials and errors. And nowadays, instead of doing the shooting in the dark trial and errors, now you can ask this job to artificial intelligence like AlphaFold. That's why AlphaFold won a Nobel Prize, because AlphaFold can predict, okay, this type of drug can perfectly fit into this type of structure. They can do this, okay? So that's why our future, along with this thing, is brighter thanks to this artificial intelligence incorporation. Okay. So same thing. So what's the difference like a DNA polymerase and ribosomal things? The prokaryotic DNA polymerase is slightly different from eukaryotic. Yes, they are doing the same job of DNA replication, but if you look in closely detailed inside, then their structures are different. So then those different structures can be attacked by different type of chemicals, and that's how you achieved the antibiotics. And this thing, how many times can you fire the replication is all dependent upon some specific DNA sequence. So on your chromosomal DNA, in the bacterial chromosomal DNA, there is a specific DNA sequence from which only your DNA replication can start. They are starting point. And they require some of the very specific ingredient to start, open up those things. Plasmid have some different kind of such things, and they allow, due to these different things, they can allow multiples of this firing, unlike chromosomal DNA. Probably only few of you can somehow agree and understand. So yeah, never mind, but that's the reason why these two different things can happen. So how do you know whether we are derived from archaebacteria rather than real bacteria, and how long time ago? It's all based upon this. All based upon this thing. How do you know whether this branching point occurred? This is a time. This time scale on this phylogenetic tree diagram is all about what? DNA sequence. That's the power of the DNA sequence comparison. The difference. You can calculate the differences in DNA sequence comparison into like indicating some times, okay, from which these two things got separated. Because always the rate of DNA change through mutation is a kind of a constant. I shouldn't say constant, but it's like along with the time. Okay. So it's a constant of the time constant, so to speak. So that's why you can accurately calculate how long time ago actually the Neanderthal and our humans were actually considered the same, share the common ancestor, versus how long time ago chimpanzee and human share the common ancestor thing, can all be accurately calculated through this DNA sequence comparison. That's the thing. Archaea and eukaryotes, are there any? If we were derived from archaebacteria rather than bacteria, and are there any some more similarity for us to like this archaebacteria? Yes, there are some. Actually, archaebacteria's ribosomes are much more closely resemble their eukaryotic things, strangely. And another thing is, probably most of you don't know what I'm saying is, but there are some things called introns. The gene element, introns. Introns are supposed to, we have known that introns are only present in eukaryotic genes. But it turns out that archaebacteria also have this introns. But all those bacteria don't have any introns. That's another very similarity things. Okay. So there are actually a lot of those similarities exist between eukaryote and archaebacteria than those prokaryotes. Actually, the bacteria prokaryotes. And here this thing is, if we abuse antibiotics, what happens is one critical damage is these antibiotics will wipe out our internal piece of internal bacteria population. The symbiotic relationship bacteria population will be wiped out because they are not antibiotics resistance, unlike, okay? So if we keep taking the antibiotics more than necessary, then all these healthy, useful, beneficial bacteria population will also get killed. And it will be a serious damage to your health. That's another really big danger that we sometimes actually get to neglect. Antibiotics, good question actually. A lot of people actually get messed up this. Vaccine, antibiotics, and also antibodies here. Antibiotics is totally different. Antibiotics are chemicals. Antibiotics are chemicals, and antibodies and vaccines are also usually proteins, and antibodies are proteins. Antibodies are defense proteins that our cell make against so-called this antigen. And these antigens are foreign molecules, usually protein. Foreign molecule, they once enter, then our antibodies recognize this thing. Oh, you are not one of us. Then anything that carries this foreign molecule, I will kill you, is how it works. What's vaccine? We are intentionally insert some benign antigens to our body so that we can have time to prepare the specific antibodies against this specific antigen is the vaccine. Okay. But antibodies are totally different. Antibodies are chemical that directly kill only bacteria. I can only finish up to so much. So it's already hit 14. So, okay. So see you later. Have a nice weekend.